our lab isolate is killing E. coli cells. And then all of these gray bars here are different environmental isolates. And you can see that there's a big range in their ability to kill E. coli. So this is almost a 6 logs difference in their ability to kill E. coli. So what I was interested in doing is trying to figure out if there is any genetic basis for these differences, or can we identify what's the genetic basis. So we didn't have genome sequence for most of these environmental isolates. So step one, like Brian mentioned, was try and sequence. So we did sequence them. And with the help of Arun, who did some of the bioinformatics analysis that I'm going to describe you now, we were able to see that there's a difference in the way the clinical and environmental isolates are related to each other. So what I'm showing you here is an average nucleotide identity plot. If you compare two different strains and they have an ANI value of greater than 95%, the convention is that we consider them part of the same species. As you can see here, all of the clinical isolates form a large clonal group. So most of them are nearly 100% identical. So they're mostly clonal. Whereas the environmental isolates are a bit more diverse and they form tiny subgroups. So the ones that are closely related form tiny subgroups. But overall, there's greater sequence diversity in the environmental isolates. Now, to dig in deeper, we decided to look at the Type VI secretion system itself that was present in all these different isolates. And so this is sort of the canonical organization of Type VI secretion systems. All sequenced cholera strains will have at least 3 gene clusters that form the Type VI secretion system. The yellow genes form the actual apparatus that's used to stab neighboring cells. At the end of each cluster are encoded different effector proteins. And immediately downstream of those are immunity proteins that protect against the toxic effects of the effector. And these structural genes are mostly conserved across strains, but the effector and immunity proteins are variable. So for example, if you zoom into one particular cluster and compare any two random isolates, you'll see that the apparatus region, and if you compare their genome sequences, you'll see that in the apparatus region there's high conservation. So they're very closely related to each other, and that's also the case downstream. But in the effector and immunity region, there's much lower nucleotide percent identity, which means that these are different effector immunity proteins. They're from different protein families. So with the help of Arun, I was able to develop a pipeline to predict different effectors since they're so different in different isolates. And this is what we were able to show. So if you look across the different isolates that we have, we found 2 classes of peptidoglycan degrading enzymes in the large cluster. We found 3 new classes of lipases in auxiliary cluster 1. And we found 1 class each of a pore-forming enzyme and a peptidoglycan degrading enzyme in auxiliary cluster 2. So these are the ones that all strains have. But we also found that some Vibrio cholera isolates have unique effector proteins that other cholera isolates don't have. So these are the ones that are not shared by all of them. So auxiliary cluster 3 was present in 9 isolates, and it encodes for this hydrolase. Auxiliary cluster 4 was present in 6 isolates. It encodes this effector protein that has an unknown function, so we don't know what the function is. And people in our lab are interested in figuring out what this is by phenotypic assays. For example, trying to compete them against E. coli or express these proteins in E. coli, try to see what the function is. I should also mention that while these auxiliary clusters have the same organization like the other auxiliary clusters, the structural apparatus genes are not similar to Vibrio cholera structural apparatus genes. They are more similar to other known plant pathogens. So this indicates that these auxiliary clusters are probably not native to cholera, but they've been horizontally acquired from other genomes. And finally, we identified a 5th cluster, Aux cluster 5, that encodes for this lipase. This is present in 2 isolates. And what's interesting about this cluster is that it lacks an easily identifiable immunity protein. So we don't know whether or not this has an immunity protein. It's possible that the immunity protein might be encoded elsewhere on the genome. And so some people in our lab are also trying to find that immunity protein. So we've been able to show that there's these distinct effector immunity proteins, but how does it matter? What do these effector immunity proteins do? How do they help cells compete with each other? So remember that we have all these diverse isolates, and so the undergrads in the lab did the painstaking work of taking each of these isolates and competing them in head-to-head competitions against each other. And the way they do this is using a killing assay. So we designate one particular strain as a predator strain and another strain as a prey strain. We co-incubate them on a solid surface at a 10:1 ratio, so predator is always in the majority. And then we plate on selective plates to measure prey survival. And using this painstaking approach, we were able to identify a pair of strains that are mutual killers. So what does this mean? So what I'm showing you here is prey survival on the y-axis. I'm competing a red clinical isolate against self, and you can see that you get very high CFU counts. But if you use the environmental isolate that's colored blue as the predator and compete it with the clinical isolate as a prey, you can see that the CFU survival, or the number of CFUs that you get, that goes down. So this indicates that the blue environmental isolate is capable of killing the red clinical isolate. If you switch it around, again, the blue predator cannot kill self, but the red predator can kill the environmental isolate, which is blue. So they are mutual killers, they can both kill each other. And the reason that they're mutual killers is because they encode different effector immune proteins. So to summarize this part of my talk, I was able to identify using genomics approaches several diverse effector proteins, and so I was able to classify them in the canonical type VI secretion systems as well as the non-canonical clusters, and then I was able to identify that these proteins are responsible for mutual killing. Moving on to the last part of my talk,